Hello and welcome to the last chapter of the molecular world, the living world biology of your NIS, chapter number 25. And today we're going to talk about the DNAs and the fundamental part of what makes them, the genes and the chromosomes. Interesting chapter, something new to learn about your own body, about your own cell. The beauty of this DNA is the very shape of it, the shape that got its scientists, whom we'll talk about, Nobel Prize. You see, it's called a double helix structure. This is how you are. It's like a ribbon which will unravel itself, and there are these bases out there. We'll talk about the ATCG bases, the four bases that makes up millions of combination of what is a basic unit of every living organism. So DNAs, if you go back, unravel the ribbon, the DNA helical structure is composed of chromosomes. Chromosomes are a part of nucleus of every single cell except the RBC. So the blood does not have chromosome, which is exactly what we say, that blood transfusion will not have the chromosome transfusion. But this is so important and you understand about it. And then before that, it's the nucleus and the cell. To understand the whole world, let's understand what a chromosome is. A chromosome, basically present in pairs in a body, it is a structure found in every single DNA. As I said, every single nucleus of a cell except the RBC. What does the DNA have? It holds the genes. A very bad trivia, but when I was a school student, and when I heard the word genes, I was really like, denims? No, it is not GENS, really bad trivia. It's the genes that make us up, the building block of human body. Chromosomes also contain protein that help DNA to exist in its proper form. Now, something very interesting about chromosomes, a bacteria may have just one chromosome. And this is what a structure of a single chromosome may look like. How many chromosomes in our body? Well, this is the number of chromosomes that we have in our body. Every single cell of our body has essentially got 23 pairs of chromosomes. Where here? So here we are looking into 23 pairs. 1, 2, 3. Out of which 23, 22 are basically same kind of chromosomes, which is what we will call as autosomes, right? So remember these small words. Autosomes are 22, and then there is an XY chromosome, which is the 23rd, which is the sex chromosome. Now, once you understand the XY chromosome, you understand so many things about the world of chromosomes. What's an XY chromosome? Well, an XY or an XX is a chromosome that determines the sex of a child. The sex of a child, how do you understand that? Well, look at it. If a mother always have these chromosomes, X and X. A father has an X and a Y chromosome. A child will have one from here and one from here. Let me change the color of it so you understand it better. So for a child to be born, who determines the sex? Which, even father doesn't determine the sex, but the chromosome from the father is responsible for the sex of the child, which is a beautiful version. If you understand science, you'll understand that the female feticide where mothers are blamed is completely unscientific. A woman can only give one of the two, which can be only an X. So an X comes from the woman, and a male can either give an X or a Y. If it's XX, then there's a, oh, wonderful, beti hui hai, the girl is born, and I'm a proud father of 3 lovely daughters. There we are. But if a father were to give a chromosome which is Y, so then you'll have X and Y, which is a boy. And trust me, it's not the pink and the blue, it's just the X and the Y chromosomes we're talking about. Fair enough? So this is the world we're living in, the number of autosomes and the wonderful world of sex chromosomes. So chromosome, essentially, the original matter of the DNA is contained in the gene. What's a gene? Well, gene are your traits, your inherited characteristics. And in the previous session, we learned about the disorders that happen because of genes, albinism, the color blindness. It's all passed from the gene, a parent gene, to the child gene. Dr. Harb bin Khorana, an Indian American. He was given a Nobel Prize in molecular biology for being the creator of the first man-made gene. I love the Google Doodle. I'm absolutely in love with the way Google says. You don't even have to say it's a Google. You can read the word Google. It's nowhere written, but this is Google, right? Is that right? Right. But this is a small tribute by Google to this man, Dr. Khorana, for his contribution in the world of molecular biology for the human gene. Who else? The first people to find the structure of a gene was by this man, Dr. Watson and Crick, and they found out the double helical structure of the DNA much before Dr. Khorana came in 1970. He came, the Watson and the Crick brothers or the friends came in 1953. There's an important name missing. And we do SDG goal number 5, gender equality, is of Rosalind Franklin. Rosalind Franklin, on the cover of Time magazine that you see, was one of the 3 scientists who discovered what is called the secret of life, that the DNA is a double helix structure. Isn't it interesting? This is a book I recommend if you're into molecular biology, you must read about it. And now a little bit about what gene theory is about. So you see these are bases. And the combination of bases gives you the genes combination, which is close to 200 billion genes that we're talking about. What are the bases? So a DNA molecule is a polynucleotide, many nucleotide, which contains a nitrogenous base, a deoxyribonucleic acid, and a phosphate. So DNA stands for deoxyribonucleic acid, right? The DNA molecule that we're talking about. What is DNA? What are the bases I'm talking about? It's A, T, C, and G. Think of ATC tower, ATCG, right? So it is adenine, guanine, thymine, and cytosine. Don't worry, it's one combination that you do, and that is how you remember your basis of the DNA. A little bit of DNA as we're doing it. Deoxyribonucleic acid is the single most important molecule in the human body. Isn't it amazing? Can I go back and do a little bit of theory with you from the textbook, as you've always been doing? Something interesting to teach you. So let me quickly go and look at it. Chromosomes and genes, we did a lot of it. We did the characteristics of chromosome, the human chromosomes, the diploid number or the haploid number. So let me quickly go. We did a little part of gene, the Mendelian factor in us. We are from a gene, the DNA fingerprinting, Dr. Khorana, the bases, and this is what we are. So name the 2 sex chromosomes. Well, isn't it difficult now? So I know it is X and Y. So we are looking at X and Y chromosome as a sex chromosome. Fair enough. How many autosomes do we have got? Well, we've got 22 pair of 44 autosomes as we talk about. Why does an organism have diploid number of bacteria? Diploid because it's one from each parent, correct? And any 2 features of a chromosome that we have, it's always in pair. The number of chromosomes are always fixed. Define a gene. Well, a gene is a segment of a DNA, right? Interesting, isn't it? A lot of things you can learn about it. Let me do 2 more. 3 steps in DNA replication. We'll talk about it. And DNA fingerprinting is foolproof. Well, we will just go through that in a little while. What else do we know about the DNA? The deoxyribonucleic acid. They are large molecules, right? In fact, in your own body, you've got 9 million kilometers of DNA in your body. Ever got tired? Well, that's all because of the DNA walking in your body, right? Interesting. If you unravel, so it's like a ribbon. So if you unravel the DNA, probably the length of it would be enough for you to stretch it to the sun and back, and again, and again, and again, and again, perhaps at least 5 to 10 times. Interesting, isn't it? And then if you were to write all the chemicals that makes up the DNA, the code and the combination, it would have 3 billion letters. Who said you're not studying enough? You are doing enough of reading, and there's an excuse not to read anymore. It can store over 10 gigabytes of information. I think Amazon clouds are overrated. Your DNAs are very important. Storage unit, the most efficient is the DNA. Well, this lady, I would have loved to ask you, find out who she is, but this is a research which says you really have your next cousin actually is a chimpanzee, because we and chimpanzee have 99.1% common genes. That's like the closest we've got. Who is this lady? You can Google Jane Goodall. Jane Goodall spent her own life working in the forests of Africa, Tanzania, particularly Rwanda, working with the little chimps, and we have amazing similarities with the chimpanzees. Right? 200 phone books. By the way, interestingly, 50% of our DNA is the same as that of the bananas. Are you going bananas the way I am going? Or I'm talking banana republic? Well, 99.9% of DNA is exactly the same as every person on the planet. Just the 0.1% seriously makes us unique. And this is about DNA fingerprinting, right? So, each and every one of our DNA is so individual, it is so specific to us that even the parents' DNA are not the same. So the DNA of individual is unique. Even a hair or a drop of blood or semen of anybody will match with only that person and nobody, which is why DNA fingerprinting is perhaps the most foolproof method to identify criminals, biometrics, and everything else that requires pure identity. Very interesting, isn't it? Fine. What are the major blood groups we've got? Remember, one very interesting concept I taught you is genes do not pass through blood. So just because you get a Captain America's blood or Justin Bieber's blood or Ronaldo's blood does not make you tall, athletic, or skillful as any one of them. So red blood cells have no DNA, which is very interesting. By the way, speaking about blood, how many blood groups do you have got? So it's very simple, right? We've got A, we've got B, we've got AB, and then we've got the universal blood group, right? A, B, and O. This is the blood group you've got. So most of us are either a combination of one of the two. The commonest group which can give the universal donor is O positive. And what happens is the genes which control the inheritance of the blood are designated as IA, IB, and I. The smaller letter I, which is the recessive gene, don't worry about it. The moment you have combination of, let's say, AB or ABI, you need to know what genes are we talking about. Just looking at it, you can say IAI, A blood group. And it's part of the question I'll teach you. But the idea is these are universal donors. By the way, rarest blood group is AB, and so much about bloods that we've got. What's plus and minus? Plus and minus are called the rhesus factor. Either you are rhesus positive or rhesus negative. Most of us are rhesus positive. Rhesus negative again is a recessive gene, a less dominant gene that we talk about it. So much about knowing who you are and about it. Can we actually slice a DNA and know? Possible. That is exactly what is known as GNA, that is known as genetic, gene therapy or gene cloning. Well, what is gene cloning is when you have diseases like thalassemia, hemophilia, which is like, your blood will not clot, or unable to make hemoglobin, which is thalassemia, or color blindness. Genetic diseases that cannot be cured except by a concept of human genome concept where genetic engineering has to happen. So how does a gene is replicated? It's difficult, right? You just can't do it. I will give you 3 easy steps for gene replication, which is easier and simpler for us. Number 1, think of it as a ribbon, right? So you have got a ribbon. What do you do with the ribbon? Here you are. Step 1 is unwinding it. So you have to unwind the ribbon. That's it. You have to understand that a gene is like a ribbon and you unwind it. It's an in-text question. It's a very common question that you get asked in your book. So I'll write. So the double helix model is unwinded. Then once you unwind it, there are new models that have to be formed. So you are forming new molecules of DNA complementary to each strand. Number 1. Once you unwind it, let's wind it again. So the third step of gene replication is winding of new one with an old thread or a parental DNA. You unwind the 2 threads of the DNA, you combine it, so now you're forming new molecule, and then you wind the old one with a parental DNA module. Not difficult, easy, wonderful chapter. Thank you so much for listening and being part of it. That ends and summarizes your entire chapter of chapter number 25. The living world is over. All the best. Go crack the exams and come out with flying scores. Good luck. Bye-bye.